Hello and welcome to B2.3, which is cell specialization in IB biology. So, firstly, what is differentiation? Okay, well, in the embryo, all cells are identical and unspecialized. But as an embryo grows, cells obviously develop along different pathways and they become specialized for specific functions. And this is very efficient instead of having all cells perform all functions. And that process is called differentiation. So you can see that in the embryo, all cells are the same, but then in an adult human, there are all of these different types of cells and many more. So how does differentiation happen? Well, it happens because different genes are expressed on different cells. And this happens through gradients of chemical signaling, which regulate gene expression. So, stem cells are cells that can divide endlessly and are either undifferentiated or partly differentiated. But the thing is, they are capable of differentiating along different pathways. So they can be used to replace lost cells. So they are very, very valuable. Therefore, cells in the embryo are stem cells. So adults have some stem cells actually, and their locations are called the stem cell niche. Examples of niches are the bone marrow, as you can see here, and hair follicles, as you can see here. So niches basically can either maintain stem cells in an inactive form or promote their proliferation and differentiation. So for example, in the bone marrow and in the hair follicles, there's continuous proliferation and differentiation, causing hair growth in the hair follicle and replacement of blood cells for the bone marrow, since these have a short lifespan, so they need to be generated continuously. However, for example, in skeletal muscle, there's also stem cells. It's also a niche, but here they remain inactive unless there's muscle injury. And most importantly, stem cells are under a lot of research because they might provide a way to generate human tissue in a lab for surgery because, again, they can differentiate to many different cell types. So there are different types of stem cells. They're not all equal, as we said, and that's because they differ in their ability to differentiate. So totipotent stem cells can differentiate into any cell type, and these are the cells found in the embryo. But shortly after, they become pluripotent, as you can see here in the blastocyst. These are capable of differentiating into a range of cell types, but not all of them. And then finally, in adults, the stem cells that you find, so for example in the bone marrow, are multipotent. So they can divide into several types of cells, but not all. So it's, as you can see here, a continuum, right? It's not, these are the three only types really, but as you differentiate, you can differentiate into less and less cells, basically. There are many, many ways in which you can differentiate, but one of them is by changing the size of the cell. So human cells have very, very different sizes. So for example, sperm cells over here are basically 50 micrometers long on average, and they're very, very narrow, as you can see, and this allows them to swim easily. Then we have egg cells, which are about 100 to 110 micrometers in diameter, so they're much larger, and they're spherical, so they actually have the largest volume of any human cell. Then on the other end of the spectrum, we had RBCs, right, which are about 6 to 8 micrometers in diameter, but only 1 micrometer thick. We'll look at that later, allowing them to pass through very narrow capillaries. And white blood cells, which are 10 to 20, even 30 micrometers in diameter. And this depends on their activation level. So the larger ones are bigger because they have a larger endoplasmic reticulum to secrete more antibodies, which are proteins, remember. Then we have neurons, which are super, super long. So even though they're only 20 micrometers in diameter, they can be even actually 1 meter long to carry signals. And finally, muscle cells, which again, their diameter is only about 20 micrometers, but they can be 300 millimeters or even more. So not micrometers, but millimeters. So very, very large cells. So the size of a cell determines its surface area to volume ratio. So basically the surface area to volume ratio, it's the surface area of the cell over the volume of the cytoplasm. As a cell grows, the ratio becomes smaller because the volume increases more quickly than the surface area. So this can be a problem. Why? Because the surface area determines how substances move in and out of the cell, right? Whereas the volume determines the rate of reactions that happen in the cell. So if the ratio is super small, substances are not able to enter quickly enough because there's not enough surface area. Similarly, waste products will accumulate because they can't leave quickly enough, and also the cells can overheat because there might be too many reactions happening and the heat cannot diffuse out through the membrane. Okay, so that's all for SL. Let's move on to higher level. So some adaptations, for example, for the surface area to volume ratio, some cells try and have a very, very large surface area compared to their volume ratio, especially if they're specialized for exchange processes. So that's, for example, red blood cells, because they transport oxygen from lungs to tissues. And the way they do this is through flattening. So they have a very flat middle. Again, we said 8 micrometers in diameter, but only 1 micrometer in the middle. And this gives them a lower volume and a higher surface area. So that's one method of adapting, so flattening of the cell. And in this way, again, they can transport more oxygen. And then we have here the proximal convoluted tubule cells. So these are found near the outer surface of the kidney, and they reabsorb most of the filtrate that's so that it's not lost to urine. There's 2 ways that they have a higher surface area. They have microvilli, as you can see here, these little hairs, and they also have invaginations, so basically inner membrane foldings. And these 2 help to increase the surface area. So 3 different ways. Remember these 3 different ways: flattening, microvilli, and invaginations. Then we also have the alveoli, which we looked at in the previous topic. And these have pneumocytes, which are a specialized type of cell. And what adaptations do these have? Well, again, alveoli are air sacs, right, where gas exchange happens. So they need a very large surface area for diffusion. So we have type 1 pneumocytes. These are the majority. They line the alveoli, as you can see here. And these are the ones through which oxygen and carbon dioxide diffuses. So they are very, very wide, but extremely thin, shortening the distance that the gases have to diffuse through, because the gases go through these walls. And then the type 2 pneumocytes, they only occupy about 5% of the alveolar surface, and they have, as you can see, a very large cytoplasm. There's another one here, right? And the cytoplasm has lamellar bodies, they're called, which secrete phospholipids. These phospholipids then line the surface of the alveolus, the inner surface. It's this green liquid that you can see here, and that's called a surfactant. So basically what the surfactant does is it reduces surface tension, meaning that the oxygen can dissolve and go out of the alveolus, but also it means that the alveolus doesn't collapse on itself. It would without the surfactant. So the type 2 pneumocytes are very, very important. Then we have adaptations of muscle cells. So again, as you can see, there's a theme here. We're just looking at different adaptations. So there's striated muscle and cardiac muscle. So striated muscle is the one that's used to move the body. So they're attached to bones, and they're made of muscle fibers, which at the same time are made of myofibrils, and they're made of alternating light and dark bands, as you can see here, and these can contract. So these have many nuclei. They're much longer and they're unbranched. And because they have many nuclei, it's actually up for debate whether they should even be considered cells. And then we have cardiac muscle, which, as you can see, so it forms the wall of the heart. And as you can see, it also has myofibrils, but it only has one nucleus. It's much shorter. The fibrils are much shorter, and they're branched. So basically, intercalated discs connect each cell with several other cells, giving it this shape that you can see here. And finally, we're looking at sperm cells and egg cells, which have a lot of different adaptations. So first, if we look at the egg cells, they have this zona pellucida over here. Not an easy name to remember, but it basically prevents more than one sperm from penetrating. It also has binding proteins on its plasma membrane, which help the sperm to fuse with the membrane, and it also has cortical granules inside, and these basically make the zona pellucida impenetrable after the nucleus of a sperm has entered the egg to prevent many from fertilizing it. They also have the yolk, as you can see here, where lipids and foods are stored. It also has mitochondria to provide ATP, and centrioles, which are needed for mitosis after fertilization happens. And then in the case of the sperm, they also have receptors in their plasma membrane for the glycoproteins in the zona pellucida. They have an acrosome over here with enzymes that can degrade the zona pellucida. They have binding proteins, which bind to the proteins in the plasma membrane of the egg, allowing it to enter the egg. And then they also have a tail, as you can see here, a very long tail, which can generate forward motion, and the mid piece over here, which has a lot of mitochondria, providing ATP to the tail. So basically allowing it to move. It gives it the energy. And as you can see here, the head, right, with the acrosome and the binding proteins, which is very narrow, and thus minimizes the resistance to movement. Okay, and then let's do some questions to wrap it up. So, which of the following is not a source of stem cells? You can pause here to think, but should be quite straightforward in 3, 2, and 1. Okay, the blood, right? So the blood is not a source of stem cells. Remember, stem cells from the bone marrow, right, generate cells that go into the blood, but the stem cells themselves are not found in the blood, whereas the embryo, the bone marrow, and the hair follicles do have stem cells. They're stem cell niches. Okay, which of the following are not characteristics, of embryonic stem cells? Well, again, 3, 2, and 1. A small number remain in the adult bone marrow. No, because the ones in the bone marrow are multipotent, whereas the ones in the embryo are totipotent. That's the big difference. And finally, what happens to the surface area volume ratio as a cell grows in size? Again, should be straightforward. I said it before. 3, 2, and 1. It decreases, right? Because volume grows at a faster rate than surface area as a cell grows. Okay, so that's all. I hope it was clear. If not, leave any questions in the comments, and I'll see you next week for the next topic. Bye, everyone.